Welcome to BIORATNA बच्चों. Our today's topic of discussion is the fruit, after which we will be also be taking up the seed from the chapter Morphology of Flowering Plants. So let us first start with the topic the fruit. What is this fruit? To understand the fruit, let us see the structure of the pistil or carpel. In case of angiosperm, angiosperm is just one of the 5 groups of flowering plants. In angiosperm, there is a structure called flower. In the flower, there are 4 whorls, out of which 2 are the reproductive organs. One is female, one is male. The female one called the pistil or carpel. This pistil, it has 3 parts. This is the ovary, then style and stigma. ये जो ovary है बच्चों, in this ovary it encloses one or more ovules. One or more ovule. जैसे आपके रूम में सीलिंग पे आप 1 लाइट लगा सकते हो, 2 लगा सकते हो, 3 लगा सकते हो. Similarly, here in ovary wall, this ovule is attached at the placenta. And this ovule, it is the most importantly, the ovule, it is enclosing the female gamete. Now, when the male gamete, it fertilizes the egg, when the male gamete, it fertilizes the egg, then this ovary, then some post-fertilization changes takes place. एक बार fertilization हो जाए, तो कुछ fertilization के बाद वाली कुछ events होती है. One of it is, this ovary, it changes into fruit. Ovary, it changes into fruit, and this ovule, it changes into seed, which is protecting the embryo inside. So, we can say that a fruit is fertilized ovary. Fruit is fertilized ovary. And this fruit is found only in case of the flowering plants. Now you know why. There is one more group, that is gymnosperm. This gymnosperm, it also has ovule. Gymnosperms also have ovule, but this ovule is not enclosed by ovary. So after fertilization, the ovule changes into seed, but as there is no ovary, so there is no fruit. ये gymnosperms क्या सोच रहे हैं? Why do I need fruit? I need a seed inside which the embryo remains protected till it gets the favorable condition. तो उसने अपना एक seed बना लिया. Now these angiosperms, they develop some business sense. What they do? They prepare a fruit around the seed. This fruit is of no use to the plant, as to say, but it is going to be very much beneficial for it. What it is providing, it is asking the animals that you help me in dispersal of my seeds and I will give you fruit, the juicy fruit in return to it. And it has helped. It has tremendously helped the angiosperms, and you can find that they can be seen, they can be found everywhere. So, the fruit is fertilized ovary, which is found only in case of angiosperms. One more post-fertilization change is that this is the ovary, and this is style, and this is stigma. Now, this ovary has ovary wall. This is the wall of the ovary. मतलब यहाँ तक आपका ये ovary है ना? What happens after fertilization? That this style and this stigma, ये यहाँ से कट के गिर जाते हैं बच्चों. This two, they fall off. When they fall off, then what is remaining? Your fruit. आपके उस fruit के ऊपर style और stigma भी थे, which has fallen off, and now there is only this fruit, which is having this wall. ये wall है ovary की, or inside there is this cavity, and in the cavity is hanging the ovule. There may be one or many ovules. तो ये जो wall है ovary की, one more post fertilization change. This wall of ovary, it changes into wall of ovary, it changes into wall of fruit. It changes into wall of fruit and it is called pericarp. This is called pericarp. So what is pericarp? Pericarp is the wall of ovary which is changed into wall of fruit. So the wall of fruit is called the pericarp. Now this pericarp, it may be dry or it may be fleshy, or it may be fleshy and juicy. When it is fleshy, then it is differentiated into three parts. The outermost, it is called epicarp. The middle one, it is called mesocarp. And the inner one, that is the endocarp. Now, when we say fruit, immediately the fruit which comes to our mind is mango. And so we think that in case of mango, it is the mesocarp which is fleshy and juicy. Epicarp uski covering hai. Endocarp, we are going to see what is a drupe. There we will see what about the endocarp of mango and coconut. But it is not always so that the mesocarp is fleshy. To understand this better, we will take the example of mango and coconut. So what you have seen about pericarp, that pericarp is the wall of the fruit. Pericarp is wall of the fruit, and when it is fleshy, then it can be differentiated into three parts. This epicarp, this is the outermost, the mesocarp, which is the middle one, and the endocarp, which is the innermost. Now this mango and the coconut. Mango and coconut both are drupes. The fruits are of different types. One of it is drupe, and this mango and coconut fruits both are developed from monocarpellary superior ovary. Monocarpellary superior ovary and has one seed. From this statement, don't think that this is the characteristics of drupe. Means a fruit has to qualify these three characters so to be a drupe. Not so. Monocarpellary means having a single carpel from the single carpel, but a drupe may be monocarpellary, and it may be multicarpellary syncarpous. So this drupe, it may be monocarpellary or it may be multicarpellary syncarpous. दूसरा ले लो, superior ovary. In case of mango and coconut, it is superior, but in case of drupe, the ovary, it may be superior or it may be inferior. Now, one seed. In this case of drupes, it may have one seed or two seeds or three seeds. So, what is common in them? These drupes, they are also called stone fruits. These are also called stone fruits in which the endocarp becomes stony. It becomes very hard. Let us see how. We are first taking the example of mango. So, this is the mango. This outermost covering, जिसे आप छिलका कहते हो, निकाल देते हो, this is the epicarp. Then here you find at the center the seed. This is the seed. Now, in between here the pulp which you eat, it is the mesocarp. It is the mesocarp. So endocarp कहाँ है? Endocarp Enclosing the seed, you find one very hard, stony covering. This hard, stony covering, which is enclosing the seed, this one is endocarp. This one is endocarp. So, this is the, in case of mango. This is the mango. So what we have seen, mango has this epicarp, mesocarp, endocarp. These three together they form the pericarp. This epicarp is membranous. This epicarp is the protective covering. This mesocarp, it is fleshy and juicy. This endocarp, it has become very stony and hard. And this is the mango. Now let us see the coconut. In case of coconut, these diagrams are there in your NCERT. So if this is the coconut, now here mesocarp है जिसे आप खा रहे हो. In case of coconut, this is the outermost epicarp. This epicarp is membranaceous. This is membranaceous. And then the mesocarp, it is fibrous. जो कोकोनट का फाइबर हम निकालते हैं, this is the mesocarp. So mesocarp is not edible. Here the mesocarp is not edible. This mesocarp is fibrous. It is fibrous. What about endocarp? The same way, this is the endocarp which has become hard and stony. So this is the endocarp, and this endocarp, it is stony. तो जो आप खा रहे हो वो आप क्या खा रहे हो? तो जो आप खा रहे हो, it is actually the endosperm. So this one, this white, this one, this which the edible part, it is the solid endosperm. Solid endosperm. और अंदर में जो लिक्विड है, जो अभी तक सॉलिड में चेंज नहीं हुआ है, दिस इज द लिक्विड एंडोस्पर्म. दिस इज लिक्विड एंडोस्पर्म. दिस वी आर सेइंग द कोकोनट. दिस डायग्राम इज दैट ऑफ कोकोनट, वेयर वी हैव सीन दैट. इसको अच्छे से ध्यान में रखना बच्चों. इसका एपिकार्प क्या है, मेसोकार्प क्या है, एंडोकार्प क्या है. फाइन. सो it is having this epicarp which is membranous, the mesocarp which is fibrous, endocarp which is stony. The edible part is solid endosperm and the liquid inside it is liquid endosperm, and it has one seed enclosing the embryo. तो एक साइड में आपको embryo दिखाई देगा. And this coconut, it has three eyes. Three eyes. This endocarp will show three eyes, which is the position of the style. Now let us study the seed. Seed, you already now know that seed is developed from the ovule after fertilization. And this seed, it has two parts. One is seed coat and other is embryo. Seed coat is protective and this embryo, it has two parts. It has cotyledons, and these cotyledons, it may be one, as in case of monocots. Monocots के जो example आपके book में है, that is maize and wheat. Or it may have two, like in dicots. Dicot example, gram, pea, bean, etc. So it has cotyledons. Cotyledon is food providing tissue. It is not going to form the plantlet. So what is the other structure? The other structure, it is embryonal axis. This is embryonal axis, and this embryonal axis, it has radicle at one tip and plumule on another tip. That's why radicle and plumule I am not going to write separately. this cotyledon and embryonal axis, when we open the seed, we can see the structure, still we can miss the embryonal axis. When we open the seed, for example, gram seed, then you will find this is one cotyledon and then this is another cotyledon, and here at the center there is this embryonal axis. This is the embryonal axis. This part of embryonal axis, this is called cotyledonous node. This is cotyledonous node. Its tip, on one tip it has plumule. This plumule is going to form the shoot system. The part between this cotyledonous node to which these cotyledons are attached and this plumule और cotyledon node के बीच का जो part है, this is called epicotyle. This is epicotyle. Similarly, the part between this cotyledonous node and this radicle, this is called hypocotyle. Hypocotyle. This seed, it has two seed coats. This is having two seed coats. First, I have shown you the open seed. This is the open seed. Now, I haven't first seen up the external structure, because external structure of seed में ही this structure is easy to understand, easy to conceive, but to understand the external structure of seed, where the micropyle should be located, what is the position of hilum, what is the position of raphe, what is hilum, what is raphe, raphe ऊपर होगा या hilum ऊपर होगा, this type of confusions students have. For example, if this is the seed I have drawn, this is the gram seed I have drawn, then अब आपको माइक्रोपाइल बनाना है. कहाँ बनाना है? माइक्रोपाइल आपको यहाँ बनाना है या यहाँ बनाना है? नाउ द हाइलम एंड राफे, क्या है हाइलम, कहाँ बनाएंगे हाइलम, वेयर इट शुड बी, व्हाट इज राफे इन दिस सीड, व्हाट इज राफे. तो दिस टाइप ऑफ क्वेश्चंस, to understand this, we need to go back to the structure of ovule. And then you will never forget, you are not going to forget what is what. So, let me show you the diagram A. Here, from the wall of ovary, this is the stalk of ovule and this is the body of ovule. This body of ovule, it has two protective coats. So these are the two protective coats. These are called integument. This is integument. This is the stalk. This is called funicle. Inside there is a complete mass of tissue. This is called nucellus. And inside this nucellus, the embryo sac will be formed, inside which the egg is there. Female egg is there. This is the ovule. Now, क्या ये ovule ऐसा ही, मतलब here it is attached to the ovary wall? Does it has to hang like this straight, ऐसे stalk and then the body, जैसे आपका पंखा होता है? No. This ovule can move, it can rotate at different angles to form different types of ovules. For example, अगर ये ऐसे है, ऐसे rotate कर जाए, आपके इक्वेटर के इसमें आ जाए, तो then you call it hemitropous. जैसे उसने एक राउंड अर्थ को दो हेमिस्फेयर में डिवाइड कर दिया. बट व्हाट इज एनाट्रोपस ओव्यूल? एनाट्रोपस ओव्यूल ही आप मैक्सिमम आप पढ़ोगे. सो इन दिस एनाट्रोपस ओव्यूल, if this is the stalk and now the ovule, it turns and then 180 degree घूम गया पूरा. So the same, the इंटेगुमेंट तो जैसे वैसे रहेंगे ना बच्चे. So these are the integument, and then this hole here, this one very important, this is micropyle. This is the micropyle. This is micropyle. पहले वो नीचे की तरफ फेस कर रहा था, now it is facing upwards. And here there is this tissue, this is the nucellus, which is having the mother cells which will form the embryo sac with its egg. Now what is hilum and what is raphe? This is the free part. जैसे ये ऐसे टर्न हुआ ओव्यूल आपका, this is anatropous ovule. जैसे ये ओव्यूल टर्न हुआ तो here the whole body was free from the stalk, funicle. Funicle से तो थोड़ा भी अटैच नहीं था because it is straight hanging. Now as it has turned, तो ये वाला जो पार्ट है जिस पॉइंट पे ये अपोजिट, ये जो फ्री पार्ट है उसके नीचे का, दिस पार्ट वेयर इट गेट्स अटैच्ड टू द फ्यूनिकल, दिस इज कॉल्ड हाइलम. तो ये हो गया हाइलम. व्हाट इज हाइलम? द बॉडी ऑफ ओव्यूल वेयर इट गेट्स अटैच्ड टू द फ्यूनिकल, दिस इज हाइलम. हाइलम के नीचे भी स्टॉक है बच्चे, हाइलम के नीचे भी स्टॉक है, and that part has fused with the body of the ovule, and this part is called raphe. Clear? What is raphe and what is hilum? Now come to the structure, external structure of the seed. तो हमने ये बनाया seed. अब इस seed में हम शो कर रहे हैं कि यहाँ पे दो seed coat है. Why? These are the two integuments and this integument is going to change into seed coat. यहाँ दो integument है, that's why these two seed coat, outermost is testa and inner one is tegmen. Outer testa, inner tegmen, both together they form the seed coat. Seed coat हो गया. यहाँ से relate भी आपने कर लिया. अब आप देखिए आपको माइक्रोपाइल बनाना है. दिस माइक्रोपाइल इज द ओनली ओपनिंग इन द ओव्यूल. तो ये ओव्यूल के, दिस इज द वाइड पार्ट एंड दिस इज द टेपरिंग पार्ट ऑफ द ओव्यूल. कहाँ पे प्रेजेंट है? ये यहाँ पे वाइड है, यहाँ पे टेपरिंग है. तो ये टेपरिंग साइड जो है, इस टेपरिंग साइड में यू विल फाइंड द माइक्रोपाइल. फाइन, माइक्रोपाइल यहीं और यहीं होना है टेपरिंग साइड में. देन here the body of the ovule, it is attached to funicle. तो इस माइक्रोपाइल के ऊपर में यहाँ पे आप शो करते हो, यहाँ पे वो अटैच हुआ था अपने फ्यूनिकल के साथ. सो दिस इज हाइलम. और हाइलम के नीचे ही होना है रैफे. इट इज द पार्ट ऑफ फ्यूनिकल बिलो हाइलम which is attached to the body of the ovule. So this one is the raphe. This is the raphe. So this is the structure of, this is the external structure of dicot seed. Thus we have seen the structure of dicot seed. In this dicot seed, बाहर से आप देखते हो, these are the two seed coats developed from the integument. This is the micropyle, the same that was present in the ovule. फिर हम यहाँ पे ये हाइलम बनाते हैं. फ्री पार्ट जो ओव्यूल का है उसके जस्ट नीचे जहाँ पे वो अटैच्ड है फ्यूनिकल के साथ, दैट इज हाइलम. एंड राफे, दैट पार्ट वेयर द स्टॉक हैज कंप्लीटली फ्यूज्ड विद द बॉडी ऑफ द ओव्यूल. तो इतना बनाते हो और आपका स्ट्रक्चर डाइकॉट सीड का तैयार. नाउ लेट अस मूव टू monocot seed. One more thing, other than this seed coat, this seed, it is having the seed coat and embryo, but other than this seed coat and embryo, some seeds may have endosperm and some seed will not have. So the one which has endosperm, it is called endospermic seed. Example is castor. And the one which does not have endosperm, they are called non-endospermic seed. And this example is gram, pea and bean. What is endosperm? It was formed during double fertilization. Double fertilization बच्चों. In case of flowering plant, सिर्फ एक फर्टिलाइजेशन नहीं होता जिसमें सिर्फ एक जाइगोट बनता है जिससे फ्यूचर प्लांट बनने वाला है. In case of human beings also, only zygote is formed. But here, एक न्यूट्रिटिव टिश्यू भी बनता है, that is endosperm. There is double fertilization. By one fertilization called syngamy, the embryo is formed, and another fertilization called triple fusion, endosperm is formed. Now in some seeds, this endosperm, it gets utilized by the growing seed, the growing embryo. In that case, the seed will not have the endosperm and it is called non-endospermic, as in case of this gram, pea and bean. Whereas in some seeds, there is so much of endosperm that it remains. So this example is castor. Thus what we have seen in case of seed, that it has seed coat, it has embryo, it may or may not have endosperm. This embryo, it has cotyledon. This cotyledon may be one, as in case of monocots, it may be two, as in case of dicots. Then it has the most important part, that is the embryonal axis. Why we are calling it embryonal axis? This is what is going to form the plant. Rest of them are helping it in the formation of the new plantlet. And now we will move to monocot seed. Monocot seed में हम देख लेंगे बच्चों कि only one cotyledon is there. Mono, one, cot, cotyledon. Now we will see the structure of monocot seed by taking the example of the maize seed. If you see the maize seed from outside, then here is the position of the embryo. This is the position of embryo. And this is endosperm. This is maize seed. But actually it is not a seed बच्चों. This is a fruit. This wall of seed, it is fused with the wall of fruit. जैसे इसका fruit है, fruit की जो wall होती है, that wall, it is dry. The pericarp is dry. So pericarp is dry and that dry pericarp is fused with the testa, that is the integument of the seed. That means this is a fruit enclosing a seed. Now we will see the detailed structure of this maize seed. What it has, this is the maize seed or the fruit as you may call it. Then here this is this big part, this is endosperm. Most of the monocot seeds, they are endospermic. One exception is orchid. This orchid is non-endospermic. It does not have endosperm. But most of them are endospermic. Here you can see in case of maize that this endosperm, it encloses, it occupies about 2/3 of the seed. This one, it is the, let me draw outside this. This is the testa which is fused with the pericarp. So this is the pericarp fused with testa. Fused with testa. Now, and demarcating the endosperm from the cotyledon, this is the cotyledon or called scutellum. From scutellum is a proteinaceous layer. This proteinaceous layer which is separating the endosperm from the cotyledon, this is called aileron layer. This is made up of protein. You have to remember it chemical composition also, name also. So this is the alluron layer. Alluron layer. And this whole thing is the endosperm. So one is endosperm. What you have seen? What? There is one seed coat. Then seed coat is fused with the fruit wall. Then there is endosperm. Then there is the scutellum. Scutellum is the cotyledon. And main thing, that is the embryonal axis. Kahan hai embryonal axis? This is the embryonal axis. Fine. So on its one side, this will be the radicle. This is the radicle, and this radicle, it is surrounded by root, cap and coleorhiza. So this is coleorhiza. And on the other side of this embryonal axis, this is the plumule, and the plumule is surrounded by this coleoptile. This is coleoptile. And this is the structure of maize seed. This is the complete maize seed. You can take the screenshot. Thus you have studied the structure of Dicot seed and the Monocot seed. I hope everything is clear. Please do write in the comment box whether you have understood it properly. If you have not understood, any doubts are there, please do write that too. Do find the link for the notes and DPP in the description box. See you in the next video with the study of different plant families, and with that we will conclude this chapter, that is morphology of flowering plants. See you. Hopefully you have liked the video. Then please like, subscribe and hit the bell icon to get all latest updates.